welcome to the 2021 National Science Conference. Today will be going over gene regulation and expression. I will be your proctor. My name is Lily Mapes, and I am an 11th grader at Mountain Range High School in Mr. Davis's Period 1 of AP Biology. Our agenda for today includes chromosomes, mutations, genetic diseases, and biotechnology. First up is going to be chromosomes, including genes and the structure and function of such. Some background information includes the human genome. The human genome is actually a complete set of nucleic acid sequences in humans. It's encoded as DNA within the 23 pairs of homologous chromosomes within you. Or other words, what makes you, you. The structure and function of chromosomes. The chromosomes, at its very base definition, is a thread-like structure of nucleic acids and proteins found in the nucleus of most living cells, and they actually carry genetic information in the form of genes, or DNA. Structure of chromosomes is actually going to be made up of DNA tightly coiled many times around proteins called histones, which actually help support its structure. The function of chromosomes is they're actually responsible for containing the DNA of an organism and instructions for genetic information. So here we have a diagram of a chromosome. You can actually see that right here we have our chromosome, and you can actually see that it's notably in the form of what you would call an X, and it's actually connected in the middle at what we refer to as the centromere. How do genes get passed on from generation to generation? It's actually typically done through a process of sexual reproduction, and it allows half of the genetic information to come from each parent. In other words, a sperm and an egg, each containing one set of 23 chromosomes. When the sperm fertilizes the egg, 2 copies of each chromosome are present, creating a full genome encoding for new characteristics, giving us 46 chromosomes total in what makes you, you. And you can actually see here on our diagram that we have our original grandmother, or that purple in that form of genes being passed down to that mother and to that grandchild. You can actually see that those genes slowly losing impact on the overall genotype of the offspring, going from 100% in that grandmother to 50 in the mother and 25 in that grandchild. And that will continue to decrease as more genotypes are contributed to the offspring. And how can we predict genes? Genes and their outcomes, either by genotype or phenotype, can be predicted through mathematical modeling, and most notably we use what is called the chi-squared formula, and it actually helps to predict the probability of a gene outcome. And so here you can see in our diagram that we are predicting the probability of the color of a Labrador's coat and the texture of such. So you can actually see here at the bottom that we're actually using what would most normally be referred to as fractions. We're actually going to be multiplying through 1 times 1 equals 1 and 2 times 2 equals 4 to create that 1/4 fraction, to actually tell us that what the genotype that we're looking for of that capital B lowercase b and capital C lowercase c heterozygous B or heterozygous C, to actually get 25% of the time, or that 1/4 of the time that'll actually occur. And what turns genes into proteins? We have transcription, translation, and expression. Transcription is actually where DNA is copied into mRNA or messenger RNA by RNA polymerase. In other words, when the DNA is unzipped and it's actually going to be scanned for what we consider to be called codons, and that's actually going to be done by the RNA polymerase to create that mRNA strand. And from there, that mRNA strand is actually going to disconnect after we go over that stop codon. It's going to disconnect, and it's actually going to leave our nucleus, and it's going to go to what we call the cytoplasm to find ribosomes. And ribosomes are either floating freely within the cytoplasm, or they're connected to what we call the rough endoplasmic reticulum. And ribosomes actually synthesize this mRNA strand to translate those codons, or those sets of 3 amino acids, into a chain of proteins. And once that's done, that chain of proteins is actually going to disconnect from that ribosome, and it's actually going to be expressed through the body. And those protein chains are going to be used to function in a certain area of the body depending upon what we need. Next up, we have mutations, or changes in the genome and its effects. Ways that mutations can occur. There are 3 main ways: substitutions, deletions, and insertions. Substitutions being substituting one nucleic acid for another, deletions deleting a base to create an incomplete codon, or insertions inserting a base to create an extra nucleic acid within that DNA strand. And you can actually see here that on our original sequence, we have that original thymine right after that cytosine, and in our point mutation, we can actually see that that thymine was changed into a cytosine, or substituted. We have an example. It's going to be the gibberellin plant hormone. Now, what is it? Gibberellin is the primary plant hormone that promotes stem elongation or length within a plant. And how does it work? It's actually mutations in the GA3H gene results in a short plant phenotype. And short stems are actually recessive phenotype for plants, and so actually that's how the gibberellin plant hormone is going to come in. So how can we describe the effect of the mutation on the enzyme? So the amino acid substitution alters the protein's shape, structure, and function. And as a result, the mutation produces gibberellin. We can actually see here that we have threonine, the THR, and alanine, the ALA. The threonine is actually supposed to be an alanine. So looking more closely at this diagram, where can we see a difference between those sets to create those enzymes? So how can we predict the change in the codon sequence and resulting in the substitution of alanine for threonine? The substitution of the adenine in the first positions of the codon for guanine, so you can actually see in that first position, we can actually see that that original alanine is actually supposed to be a guanine. And how can we describe one heterozygous and two homozygous copies can have the same phenotype? And it's actually because enough active enzymes of the gibberellin protein are produced by the presence of only one wild-type dominant allele for the gene. So there actually doesn't need to be much impact or presence of the gene in the genotype for that to occur. So how can the role of meiosis play in mutations? So the role of meiosis is typically done through creating offspring in an organism. And so if a mutation occurs in cells that will make gametes through meiosis, it can actually be passed on to offspring and contribute to the genetic variability of the designated population. So those 4 daughter nuclei, if a mutation occurs, all 3 of those will actually have a mutation, and that can actually increase the likelihood of it being passed on. Next up, we have genetic diseases, or diseases brought on by mutations. Today, we'll be specifically looking at what is referred to as Down syndrome. 6,000 infants in the U.S. per year are born with Down syndrome, and actually 1 in 700 babies worldwide have the mutation. Mutations around the world, we can actually see that Down syndrome is most common in places such as Argentina, Russia, and Australia. Now, Down syndrome is actually 95% of the mutation is caused by what is called trisomy 21, and it actually affects the 21st chromosome within a genome. And it causes 3 copies of the chromosome 21 instead of the usual 2. And it's actually done caused by abnormal cell division, and it's during the development of the sperm or egg cell. And the other 3% of Down syndrome cases are actually caused by what is translocation. Now, what are the phenotypic symptoms of Down syndrome? We have flattened face, a short neck or stature, small hands and feet, or almond-shaped eyes. Next up, we have biotechnology, and technology used to study genes. Now, how do these two relate? Genetic engineering, also referred to as genetic modification, is the manipulation of an organism's genome using a system of biotechnology. And it's actually a set of technologies used to change the genetic makeup of cells, including the transfer and placement of genes across boundaries within a species or within 2 different species. We're actually going to look particularly at what is known as ischemic heart disease. And ischemic heart disease, they actually did a study, and it included 117 patients and 991 DNA samples. They actually used and studied plasma lipids and how these two related. They used blood collection for genotyping, DNA isolation, and detection of SNPs. And they actually studied the genes associated with lipid metabolism, and they were actually able to replace or manipulate the genes into actually causing a variance in the ischemic heart disease in offspring and in patients as well. Thank you so much for coming today. I really thank you for coming and listening to me. Thank you for the support, and those are my credits. Have a great rest of your day and keep learning.